topic of our class will be it's like Suraj ko chirag dikhana. I will be teaching you something which you believe you know, and that's quite tricky. I mean, teaching somebody which he or she already knows. It's very weird. And the topic's name is polymerase chain reaction. And the polymerase chain reaction is very famous nowadays. Almost 60 to 70% of Pakistani population, 100 million people or more than 100 million people now knows about the term PCR or polymerase chain reaction, thanks to the pandemic, that everybody get a sense that there is a test called polymerase chain reaction. मुझसे एक आसान सवाल पूछने दो आप लोगों से. What is the polymerase chain reaction? I'm sharing my screen as well with you. Let me see. कौन है बहादुर? किसने माइक अनम्यूट किया? शाबाश. Tuba. Is Tuba?
Yes.
Can you see the screen?
Yes, sir. PCR reaction is a reaction in which you use it to amplify a segment of DNA.
Shabash. Is to amplify? Is to amplify?
Segment of DNA.
Shabash. Good. Anybody else? Anybody else wants to add anything which Tuba has told us about the PCR? Hamza, ji.
Sir, PCR reaction is basically a photocopy for genomic information, hereditary.
हमजा आप माइक के पास बोलिए जरा माइक के पास बोलिए माइक्रोफोन पास कर लें थोड़ा सा.
I'm saying that a PCR is basically a photocopy for genomic information or nucleic acid information.
Actually, I'm not getting your answer clearly. Just try to speak once again. I will try to get it. Okay, your voice is not coming well to me. I'm not sure if the other people are getting your voice clearly or not. Ji.
Can you hear me better now, sir?
Yeah.
I said that a PCR reaction is basically a photocopy machine for hereditary information. For a sequence of genes, we can use it to make multiple copies of that specific part that we require.
Shabash. Tuba and Hamza says that it's about amplify a particular DNA, piece of the DNA, or photocopy, like a photocopy machine. We can multiply a particular gene or particular piece of DNA. So that is something we know. Anybody, if somebody has something more to tell me, he or she can. Otherwise, this is something I am taking it as a definition of polymerase chain reaction. Okay? Ji Shaista.
Sir, we take small sample of DNA and then amplify it to make a large amount of DNA.
Okay, using a small amount of DNA, we able to have more DNA. That is another definition of PCR. That's the school.
Yes.
Okay, shabash. Okay, since I'm not कोई और बहादुर है? Eman नहीं कोई नहीं है. Eman ने off कर दी. और कौन है? Okay, so let me create the question. You people needs to focus on the question, okay? And then answer को हम फिर देख लेंगे. So in order to create the question, let me share my screen once again. In a PCR reaction, if you can see my screen, okay, in a PCR reaction, we able to successfully amplify a particular portion of the DNA. That's what we can do in a PCR reaction. What is the mechanism? What are the components involved? We will be studying everything, okay? अभी हमने बिल्कुल नहीं पढ़ा है. I'm just trying to make a sense of PCR reaction in your mind before we will study that topic formally. So this is a double-stranded DNA. We want to amplify a certain part of that DNA, and this is the place. This is from here to here. We want to amplify that portion of the DNA, and then after the PCR reaction, what will happen? We able to amplify that part, and then we able to like have like 2 copies and something like 4 copies and then like 8 copies and then like many copies, and in 20 more cycles we ended up like 2 million copies of that piece of the DNA. That is the most, I must say, the most misunderstood phenomena on the planet Earth about the PCR reaction. My question is that when we want to amplify a certain portion of DNA, please stick with the question, okay? All of my class, this is going to be a very important information for you, okay? This will like a difference. I know it's quite difficult, especially in an online class, to keep your attention. I wish पहली दफा मैं अपना क्लासरूम मिस कर रहा हूँ. I desperately need a whiteboard. I desperately need that eye-to-eye contact to explain you this, okay? Because I believe that if you are like my student, you should be able to understand that concept very clearly. So let me phrase my question. If I want to amplify a certain portion of DNA, for example, if I want to amplify, this is a piece of DNA, and I want to amplify from here to here. I want to amplify that part of the DNA. At the end of the PCR reaction, I will be able to have millions of copies of that portion of DNA. Let me mark that portion of DNA like this. So after PCR reaction, I will be able to get that part of the DNA, millions of copies of only that part of DNA. I will never be able to get any DNA from this side. I will not be able to get any DNA sequence from this side. I will only able to get the specific DNA which starts from here. It's a start point and it ends here every time. So this will be the start sequence, start point, and this will be the end point. So every time whenever I do PCR reaction, I able to amplify that portion of the DNA only. This is called specific amplification. Remember that term. This is called specific amplification. My question is that why and how it is possible that the mechanism of PCR gives you specific PCR product. How and why the mechanism of PCR give you this specific PCR product? Because somebody wants to answer, please.
I think so, but I don't really remember much, but I think because we use sequence specific primers, and since they're complementary to a specific sequence, so they'll bind to a particular sequence, and then the polymerase is going to ensure that the sequence continues henceforth.
Okay, Zainab, thank you very much, and shabash to me, because I'm exactly expecting the same answer from the students, that the sequence specific primers allows the amplification of DNA for a specific reason. it seems very much possible if you understand the PCR in this way, and that is how most of us understand the PCR reaction. एक प्राइमर यहाँ लगता है, एक प्राइमर यहाँ लगता है, वो इसको एम्पलीफाई करता है, ये दो कॉपीज बन जाती हैं, and that is where I'm sorry, that is where the difference between a chemist and a biologist exists. And you are unfortunate that I have master's as well as an MPhil degree in chemistry. This is the template double-stranded DNA. I'm going to share you with the mechanism of DNA. Try to imagine what is happening within the tube when we perform a PCR reaction. This is the template DNA, for example, from a bacteria or from human. It is supposed to be a very long thread of double-stranded DNA, and we are not interested in any sequence outside our target sequence which we want to amplify. So that means we are not interested in this portion and we are not interested in that side as well. And also, you know, based on my earlier talk, that only that green portion is amplified. Are you getting me? Are you with me? Only that green portion is amplified, not other portion is amplified. In order to understand that thing, I am sharing with you the mechanism of polymerase chain reaction. So at the first, DNA is present in the double-stranded state. The green is the specific portion, the pink, blue, the other things are non-specific. In the first stage, we boil the DNA almost at the 95 degrees centigrade. This is called denaturation. And this allows the double-stranded DNA to become a single-stranded DNA. The hydrogen bonds are gone at that temperature. Then we lower the temperature to about 60 degrees centigrade. That allows the primers, the specific primers which we have designed, allows those primers to anneal themselves at the place of their choice. There will always be two primers. The first is called as the forward primer, the other is called as the reverse primer. And those are the specific primers. You can see if the sequence of the DNA is TAG like this, the sequence of the primer will be ATC, complementary to that one. It will be complementary to that one. Similarly, if the sequence of the DNA here is GGACTGC, the sequence of that reverse primer will be like CCT, it's complementary. And this specific sequence are supposed to be the reason of specific amplification. That was the general understanding. But the problem with this understanding is that, if this is true, then let me take you to the next phase. After the primers anneal themselves to the specific places, the DNA polymerase will come. These are the DNA polymerases, this one and this one. And you know, we need two DNA polymerases. And we have two strands. That is why we need two DNA polymerases. One of the primer will bind itself to one of the strand. For example, the forward primer will bind itself to the antisense or nonsense strand. Then the reverse primer will bind itself to the sense strand. And then the DNA polymerase will start making the new strand of the DNA from 5 to 3 side. That is something which you should remember. The DNA polymerase synthesized DNA from 5 to 3 prime site. Similarly, this primer will bind itself to the antisense strand or the nonsense strand, and DNA polymerase will start making DNA from 5 to 3 sides. So that is how the DNA will be synthesized. These little things are the dNTPs, the nucleotides. These nucleotides will be incorporated by the DNA polymerases to make the new DNA strands. So there will be 2 DNA strands will be created. Just follow the DNA polymerase. This will incorporate another one, another one, another one, another one. The process will continue. The process will continue. The thing where you should bang your head in the wall is that the DNA polymerase do not have eyes. It should kept on synthesizing the DNA. This DNA polymerase should kept on synthesizing the DNA because there is no stop over here. There is no speed breaker here. Nothing is going to be stopped here. It's a chemical reaction. An enzyme is there. The DNTP is there. The conditions are there. So it should kept on incorporating the nucleotides even after the specific portion of DNA. Similarly, this DNA polymerase should kept on incorporating DNA even at the end of the specific DNA. There is nobody here. There is no signal. There is no machinery which can let the DNA polymerase know, oh, the specific place is stopping. The specific place is done here. Please stop. No. So it will kept going, and kept synthesizing until unless it is stopped by the next cycle. It will kept synthesizing the DNA in this direction. Nobody can ask this DNA polymerase to incorporate the DNA here, to incorporate the base pair here, but please incorporate the base pair here, but please no DNA, no base pair should be incorporated after that. Because in whatever the principle the DNA polymerase is using, those principles are applicable here, 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 and applicable for the rest of the molecule as well. So many people have a very weird understanding about the polymerase chain reaction in their mind. And that weird understanding is that the specific primer will start synthesizing the DNA. One primer binds here, the other primer binds here. They start making DNA and they collide with each other, something like that. And then they make a double-stranded DNA. It never happens like that. They got separated. There are 2 strands. They independently incorporate their own primers. They independently catch their primers. And then once the primer will start incorporating the DNA, there is no way that the primer should stop here. It will kept on synthesizing the DNA. So there is no way that after the PCR reaction, those specific products should be here. The specific products. There should be products which are like longer ones, because who will tell the DNA polymerase to stop here? In pictures, in drawing, it makes sense. But in the real chemical reaction, when that blue primer, if I can zoom this one for you. When that blue primer, oh my God, that blue primer will start synthesizing the DNA. The template DNA is not stopping at that stage. It's supposed to be here. It's supposed to be here as well. So this will kept on synthesizing the DNA until the template DNA is not finished, or the conditions should not are same. Similarly, that primer will kept on synthesizing the DNA. It will not stop here. Nobody tells that primer, please do not, or the DNA polymerase, do not incorporate any more base pair after. It will kept on putting more base pairs. So the concept of specific amplification, the specific amplification is there. Let me tell you, it is there. But it is not because of the primers, because the primers will not be able to end at the other side. It can start from a specific site like this one. It can start from a specific site, but they cannot end the DNA synthesis at this site. Similarly, it can start synthesizing the DNA from that side, but it cannot end the process of synthesis at that side. Am I clear in my question? Zainab, are you getting the point? So the thing which you are having in your mind is not the real answer of specific amplification, because the primers cannot stop the synthesis of DNA at the other side. If anybody want to make any comment at that time, please speak. If you want to, if you believe that it is fine, understandable, or if you have another point, another explanation that why a specific amplification occurs during the polymerase chain reaction. Using the chat box, this is the most unkind thing I allowed you people to do to me. So the part of DNA, if we are amplifying a gene, it have a start and stop codon to the PCR can end when the polymerase will stop the reaction over there. I'm not speaking the name as a promise. Oh, my dear student, I'm not speaking your name. The start and stop codon stop the translation procedure. This is not translation. This is transcription. And DNA polymerase do not have any idea about the start and stop codon. It actually start making the DNA from any place. It start having a reaction. The concept of start codon and stop codon is for the translation, for the incorporation of amino acid. ठीक है? Since मैंने तेरा नाम नहीं लिया इसलिए I can use this thing. Okay? शाबाश. अगला बोलते क्यों नहीं हो? यदीश बोलता क्यों नहीं है? You might be a YouTube star, okay? Sir, with each cycle its length shortens. Who? कौन करता है? यदीश बोल के बताओ. कौन छोटी करता लेंथ? कोई चीज है मैकेनिज्म में जो अगले साइकिल में आ जाएगी? जो पहले साइकिल में पीसीआर की ट्यूब मशीन में जी शाबाश.
कंफर्म नहीं है, मुझे लगता है मेरा आंसर गलत ही होगा, लेकिन चले. आई गेस
you पहली बात कृदेश, no worries about the wrong question, okay? Chill.
Yeah.
शाबाश.
सर आई गेस जब वो डीनेचर हो जाता है तो उसपे फॉरवर्ड और रिवर्स प्राइमर जिस पॉइंट पे लगते हैं तो उसके पहली बात तो उसके पीछे वाला हिस्सा तो वो डिलीट हो गया ना साथ, क्योंकि आगे एम्प्लीफिकेशन उस पॉइंट से आ रही है.
ओके फरहान,
और
बोलता क्यों नहीं है तू? अच्छा कृदेश अभी आप बोल रहे हैं. सॉरी प्लीज कंटिन्यू.
और फिर अब जब वो क्योंकि जब टेम्परेचर नीचे आएगा वो डबल स्टैंडर्ड बन जाएगा. अब फिर जब वो सिंगल बनेगा तो फिर उसमें ऑपोजिट प्राइमर जब आके लगेगा वो आहिस्ता आहिस्ता उसकी ऐसे लेंथ शॉर्टन होती जाएगी. आई गेस.
गुड शाबाश ठीक है. आई मीन ठीक है मींस यू आर फाइन विद योर पॉइंट्स. गुड. हम इसको पकड़ लेंगे, ढूंढ लेंगे. शाबाश. और खुद ब खुद ही आपके सवालों के जवाब मिलना शुरू हो जाएंगे. ओके फरहान बोलेगा नहीं तो मैं बोले मुझे बोलना पड़े. सॉरी मैंने आपका नेम ले लिया पहले यार. इट विल ओनली हैपेंस, क्या पता मैंने तुझे स्कोल्ड करना हो क्वेश्चन में. It will only happen in the first cycle. The polymerase will have a limit of integrating a specific amount of bases before falling off, but in the next cycle, the primer will have an end point that had been made by the previous primer in the first cycle. Okay, I will show you what happened in the second cycle. You came to know. You are somehow close and with an idea, and I'm happy, but it's not, still, we are not there. I mean, remember, the capacity of the DNA polymerase will not be changed. If an enzyme is working today in a certain conditions in one way, if you repeat the condition, it will start working in the same way. There is no falling off. The template is there. It will keep on synthesizing that DNA. So that's fine. अगर एक strand खत्म हो जाती है, that is another different thing. ये थोड़ा सा मेरे मतलब की बात है, and we will see the clear मतलब soon. Anybody else? I have another point. Okay, another question. Shabash, please keep making your comments. That's fine. अगर आप बोलेंगे तो इतना अच्छा लगेगा यार. Okay. So let me maybe due to DNTP if we add. देखा DNTP जो हमने ऐड की है ना, वो पहले साइकिल में भी मिक्सचर में मौजूद है, फिर वो अगले साइकिल में भी मौजूद है, फिर वो अगले साइकिल में भी मौजूद है. तो सारे साइकिल्स में DNTP, जब DNTP खत्म होगी, the polymerase chain reaction will finish. I mean, every type of DNA synthesis will stop. So what that means, that we should not have a specific DNA, we should have a mixture of different lengths of DNA. लेकिन PCR में specific product बनती है. जी, शाइस्ता आप कुछ कहना चाहती थी? माइक्रोफोन आपने किया जी।
सर मे बी डीएनए पॉलीमरेज के स्ट्रक्चर में कुछ चेंजेस आती हैं जिसकी वजह से वो स्टॉप कर देता है सिंथेसिस।
नहीं ऐसा पॉसिबल नहीं है, बिकॉज पीसीआर ट्यूब के अंदर डीएनए पॉलीमरेज एक एंजाइम है. इफ द डीएनए पॉलीमरेज चेंज इट्स स्ट्रक्चर, दैट मीन इट विल चेंज इट्स एबिलिटी टू सिंथेसाइज द डीएनए एज वेल. सो द एबिलिटी ऑफ द डीएनए पॉलीमरेज विल स्टे सेम. ठीक है, वो उसमें कोई फर्क नहीं पड़ता. ये भी पॉइंट. शाबाश शाबाश, गुड गुड. एंड डोंट वरी if your answers is not ended up correct. Okay, don't worry, just keep on sharing your mind. That will help you to clarify your concept in a different way. I'm happy with all those points. I just want enough time so that I will be able to finish the answer as well. Or online classes may, I mean, you people have all the choices. But I know there are many brilliant minds out there who are actually googling that question, and thank you very much that one as well. And I want to get that answer. Okay, so I will going to the. Okay, anybody wants? जी हमजा, आप कुछ कहना चाह रहे हैं? जी जी शाबाश.
सर जब हम एक स्ट्रैंड लेते हैं, लेट्स से सेंस स्ट्रैंड, और उसको कॉपी करते हैं, तो ऑफ कोर्स इट इज ट्रू कि द डीएनए पॉलीमरेज विल नॉट स्टॉप एट अ रैंडम पॉइंट. मतलब एट अ स्पेसिफिक पॉइंट वो रैंडम पॉइंट पे ही स्टॉप करेगा जहां पे हमारी साइकिल एंड हो रही है. लेकिन जो इसकी कॉपी बनती है वो इसकी एग्जैक्ट कॉपी नहीं होती, वो इसकी कॉम्प्लीमेंट्री कॉपी होती है. सो द नेक्स्ट प्राइमर दैट विल अटैच विल बी द रिवर्स प्राइमर, right? And the reverse primer will copy only until the start of the forward primer.
Okay.
And that is why we will only have a specific length of the gene after the second cycle.
Okay, I'm taking your point. Let me it's all are wonderful. Okay, so let's see the procedure. Okay, how actually polymerase chain reaction proceed. आपने एक बात पे ध्यान रखना है. You need to focus on the product of every cycle. Okay, अपने mind में notebooks पे कहीं पर भी कि what we are getting at the end of the cycle. Each cycle, at least for first few cycles for sure. It should be very clear. So at the end of the first cycle, which I showed you, we will be having the two products. Am I right? Two products. None of them is specific. ठीक है Hamza? And the other people? None of them is specific. So that means there will be no PCR product. Specific products are produced after the at the end of the first cycle. तो ये मिथ गलत है कि पहले साइकिल के एंड पे 2 कॉपीज होंगी. पहले एंड के साइकिल पे how many specific copies are there? The answer is 0. No specific copies there. But now we have 2 templates. Am I right? Actually, we have 3 templates. Shabash, somebody wants to answer? We have 3 templates now. At the start of the third cycle, we have 3 templates. Ji, agar the the 2 templates you are looking at, the 2 new products, and the third template is the original one, the original DNA. जिससे हम लेकर चले हैं वो भी तो है, जिससे हमने डीएनए सिंथेसिस शुरू की है. द ओरिजिनल टेम्पलेट इज देयर एज वेल, और वो बहुत सारा है. इट्स टू मच, ये तो अभी 2 हैं सिर्फ, जस्ट 2 कॉपीज. ओके, लेट्स मूव टू द थर्ड साइकिल. सो इन द थर्ड साइकिल the cycle will be repeated. You know how it will be repeated? First we boil the DNA almost. Now these are the 2 products we are having. Can you see the 2 products? They are a little bit weird because the primer binds here, it start producing the DNA. It will not stop here. Kept on going. क्या नाम रखें इस प्रोडक्ट का? इसको मैं नाम दे दूं unspecific product. It's a non-specific product, something which we don't want. It's a non-specific product, or we can say it's like elongated non-specific product. And this one, another primer binds here, and DNA synthesis from this direction. It kept on going, not stopping here, kept on going. So we will get 2 products, both of them non-specific. And do not forget the original template, it's still there. Original template कौन सा था? ये वाला, ये नहीं, ये वाला. We have the original template as well. So all these three will enter into the second cycle. At first, the double-stranded DNA will be single-stranded DNA, and then the process will repeat itself. We have three templates. If the original template is used, what will happen at the end of the cycle? These two products will be produced. Usi tarah, if these two templates are used, then what will happen? We will going to see that. So we have three templates once again. If the original template is used, what will be the products? These two. If these two are used as a template, what will happen? I'm going to show you. So what will happen? 95 degrees centigrade, these strands will become single-stranded DNA, and primer forward and reverse will come and try to bind its target locations. Do not forget the target location. One of the primer binds here in the same way. The other of the primer binds here. The color is same, so it is difficult to pick this one. Similarly, one of the primer binds here, the other primer binds here. Everything is clear? Now let's proceed after the annealing DNA synthesis. Now this DNA will start synthesizing from this direction to this direction. It will not stop here. It will keep on going in the same way. What about this primer? This primer binds here, will start making DNA, but it will end here. Why it ends here? Because there is no template after that. It will stop here. Think about this one. This primer will bind itself here, will start making DNA, and will stop here because there is no template after that. Think about this primer. It binds here, start making the DNA, will not stop here, kept on continue. So all these four will undergo in the PCR reaction and will going to produce the four new products. Imagine the four new products, how these four products will look like. DNA polymerase will come, start making the process of DNA synthesis. Full stop. These are the four products. Now we have four longer molecules. I mean, these are not specific. So at the end of the second cycle, if somebody asks you how many specific PCR products will be there, what should be your answer? Zainab, please speak. What should be your answer? At the end of the second PCR cycle, how many PCR products will be there? PCR specific products. G?
There are two, I guess.
There is no PCR product, no specific PCR product is there. The specific PCR product should be in green.
Yeah.
So we have no specific PCR product yet. ठीक हो गया? Now can you tell me how many template we are having at the end of the second cycle? The four products you are looking at, the two products at the first cycle, and then the original DNA. Do not forget the original DNA is there as well. 1 and 2 and 4. That makes 7 templates, different types of, not different, 7 types, 7 templates are there. So at the end of the second cycle, there exists no specific PCR product. तो वो लोग जो कहते हैं ना दूसरे साइकिल में 4 बन जाते हैं, वो एक्चुअली गलत कहते हैं. जो कहते हैं पहले साइकिल के एंड में 2 बन जाते हैं, वो भी गलत कहते हैं. दूसरे साइकिल के एंड में 4 प्रोडक्ट्स बनती हैं, वो भी गलत. नो प्रोडक्ट इज फॉर्म्ड एट द एंड ऑफ द सेकंड. हाँ, 4 वियर्ड सी लॉन्गर मॉलिक्यूल्स बनते हैं, but they are not the things which you want to prepare or the things which you are intended. Should we move to the third cycle? That will create a mess. चले आगे, let's move. These 4 products will enter into the third cycle. But let me help you just to revisit the earlier things. We have 7 types of templates, am I right? If we use the original one, we are nobody who will decide to use. It's in the machine, it's in the mixture. DNA polymerase don't have eyes, it's a chemical reaction. So if the chemical reaction choose to start with the original DNA, what will happen? कौन सी products बनेंगी? Which you saw at the end of the first cycle. If the chemical reaction choose to start with the products of the first cycle, what will be the products? The 4 which you see in the last cycle. If the chemical reaction choose to start with the 4 products which you saw in the second cycle, what will happen? We will be looking at right now. So these 4 products will go into the third cycle. Denaturation, the double stranded will become single stranded. The primer will find their places. One of the primer will bind here. Remember, this is the green area. And the other primer will bind here. One of the primer bind here, the other one will here. If you want me to show you the 4 products, look at the 4 products once again. These are the 4 products which entered into the third cycle. This double stranded DNA will become single stranded. This double stranded DNA will become single stranded. They got separated. See how? Once again, these 4 products, end of the second cycle, cycle number 2, start of the third cycle, the 4 products will generate 8 single stranded molecules. The primers will be incorporated. Here comes the primer. There should be some sparkles in your eyes now. When this primer starts synthesizing the DNA, it will not stop here. It will kept on going. It will produce what? The longer non-specific thing. What about this primer? If this primer kept on synthesizing DNA, it will end here because there is no template. And what about this one? If this primer start making the DNA, it will stop here because there is no template after that. And what is that? This is the specific PCR product we want to amplify. So in a nutshell, this is the third cycle where you actually saw first time the specific products. All other are non-specific. एक यहाँ बनेगा, the other one will be here. So at the end of the third cycle, we will be having like how many products? 1, 2, 3, 4, 5, 6, 7, 8. And out of these 8 products, only 2 are specific. And 6 are the longer non-specific products. Now, what was my question? What factors defines the specific amplification? Just keep looking at the mechanism. We found 2 specific products at the end of the third cycle. How about the next one? Let's go. Are you ready to go to enter into the fourth cycle? It will be a chaos. Hai? It will be a chaos after that. Okay, so we have longer products here. We have the specific products here. All of them entered into the fourth cycle. The double stranded DNA will become the single stranded DNA. All these products will become single stranded DNA, the specific ones as well. The primer will find their places and the DNA will be synthesized in the same way. So at the end of the fourth cycle, these 2 specific products will amplify and we have 4 more specific products at the end of the fourth cycle. So we have 8 target molecules along with the 8 longer length molecules at the end of the fourth cycle. So even at the fourth cycle, most of the people believe we have like 32 molecules, something like 16 molecules. The answer is no. We have only 8 specific products. If these go into the next cycle, all of the specific ones definitely we're going to produce the specific products, and we get more specific from there as well. So all these specific PCR products will create more specific products, and we get more specific from these as well. So at the end of the 5th cycle, we have 22 target molecules and 10. So you can see that specific products are increasing in number exponentially. And at the end of the 30 cycle, we have like 1 billion specific molecules and only 60 longer length molecules remain. They left so much far behind after few cycles. And after 6, 7 or 8 cycles, the DNA polymerase will fail to find the longer length molecules because they are too less in number. All the DNA polymerase and DNTPs will involve in the specific products. So when you load that PCR product on the gel, you will only able to see the specific product. So this mechanism, if you understand that mechanism, is the only way to understand that why a specific PCR product is created during a polymerase chain reaction. Of course, there are other factors which we use to help to amplify the specific PCR products, and I will be discussing those factors and the mechanism of PCR reaction in the next lecture, in the next class. If you understand the question well and you're able to understand the mechanism of initial cycles, you will be able to understand that why specific amplification occurs during the polymerase chain reaction. If you have a little love with astronomy, you might heard or know that people always want to know about how how universe actually came into existence, the Big Bang. They try to create simulations to know what happens in the initial phases immediately after the Big Bang, because that will set the tone what will happen after that, the galaxies and the stars and all these things. The initial cycles of the PCR reaction defines that how the PCR reaction will proceed. If you understand the mechanism, I can tell you, you will be able to control your PCR reaction in a very, very efficient way. You can troubleshoot the things more easily. You can figure out why your PCR reaction is actually not ended up in a success. Let me answer you a very important question before I conclude the session. And in the next session, I will be taking your questions as well together with the mechanism of PCR, because it's very easy. The difficult stuff is what we have studied. The people believe when they are doing PCR reaction, if you happen to be there in a certain lab, they add a certain amount of template DNA in a PCR mixture and they fail to find any result, and just they start making changes in their protocol. They add more template. They assume that adding more template will help them and give them a better chance to get the PCR product. And if you understand the mechanism which I taught you, adding more template is a curse for the PCR reaction. You know how? If we have an original template in huge numbers, the DNA polymerase will choose to bind to the original template. If the DNA polymerase will choose, if the primers will choose to bind with the original DNA. To get the specific amplification, it is important that the PCR primers should bind to the products of each cycle, not to the earlier ones. You starting with the original DNA, you get the products. Now you want the primer to bind with the products as rather than the original DNA. If they bind with the product, they will move to the next cycle. If they bind with the products of the next cycle, cycle number 2, they entered into the third cycle, where you start looking at the products. If they choose to bind with the products of the cycles, further cycle, they will enter into the next cycle to produce more specific products. If the original DNA or the original template is in a huge concentration, the primers will be consumed in binding themselves with the original DNA. When they bind with the original DNA, they will produce what? They will produce the products of the first cycle. That means non-specific product. And you are not looking for the specific non-specific product. So when people do the things, ज़्यादा चीनी डालो, ज़्यादा मीठा होगा, template और कर दो, PCR में product आ जाएगी, they usually fail to find the product because of this technical misunderstanding they are having, or less knowledge about that mechanism. This is just one example I am telling you, which can help you to troubleshoot your PCR. I will be discussing more troubleshoot factors. If you understand the mechanism, you can tune, you can guess what's going on. You can guess what's going on within the tube. I hope this will help you, and I am left with like 1.5 minutes of the class, and I really hope, and I'm hoping that मैंने सूरज को चिराग दिखाने में कोई कसर छोड़ी है या नहीं छोड़ी है. If you are feeling comfortable about the PCR or feeling different about the PCR, I will be happy as a teacher that you are getting things about the PCR in a different way. Please keep asking your questions in the upcoming sessions. Okay, so before you go, let me complete my ritual. Okay, so please stay safe and stay healthy, and try to be very careful in the upcoming days.